So almost from the earliest time that we started to use antimicrobial drugs, there has been an acknowledgement that because what we are doing as using these drugs against other living things, the capacity of other living things to adapt to the threats just in the same way that we can has always been there. And the drug resistance hasn't just started happening, it has been happening throughout the history of our use of antimicrobials. The problem now is that we're getting to the point where almost all of the antimicrobial drug classes that we have are starting to be less and less effective against some of the leading disease-causing bacteria. And that's the problem. We're having to fall back to more and more toxic drugs in order to actually address some of the bacterial infections that used to be really no problem to treat. And we're having to use combinations of drugs, and we're getting increased numbers of deaths from infections that didn't used to cause those same problems. So you can see a number of public health campaigns here. Well, first of all, raise awareness and try and address the problem in some way from a public health perspective. So this one here, this little lad is trying to discourage the use of antimicrobials, they're using the term antibiotics, against viruses, because they don't work against viruses, and there's no point in giving people antimicrobials if they've got a viral illness or if you suspect they have a viral illness. And this little bit of advice down here is getting into a bit more detail about that same kind of thing, is that don't just hand out your antimicrobials, have a look and see what it is, positively identify what the microbe is first, then when you give an antimicrobial, then you'll be very careful about the dose and the course, and the course is properly taken and so on. You could see here at the bottom with methicillin-resistant Staphylococcus aureus infections on the skin. It's a great big crater in the skin, which is the kind of thing that you see with Staph aureus infection. You verify that it is MRSA before you try to treat it, and you don't use vancomycin, which is the last resort in antimicrobials. You don't use that if you don't have to. So you've got to find out first that it is resistant to everything else. It's resistant to methicillin, which is the next from last resort, before you go to the vancomycin. So various bits of advice about how to use antimicrobials better, but also a lot of discussion amongst healthcare providers and researchers about how to combat drug resistance. And this little thing here is giving us some clues as to what a lot of the problems are. Irrational drug use, that's not just in medicine, but also in food production and various other areas. The use of antimicrobials has been widespread and in some cases doesn't make any sense. Lack of research, we'll talk about that a bit more in a minute. Weak surveillance about exactly what's going on and who's got what infections and how resistant they are to drugs. Poor infection control, that is how to actually prevent infections being transmitted from one person to another by just doing the general sort of cleaning up, particularly in hospitals and other healthcare facilities, preventing there being infectious organisms allowed to survive generally around in the environment. We referred to that with Clostridium difficile, that's that bug that causes pseudomembranous colitis. The incidence of those infections is rising. That's a bacteria which can survive for quite long periods of time outside in surfaces, particularly if they're relatively moist, and so infection control for those types of bacteria is particularly important. You really have to go back to your protocols about how do you clean, how do you disinfect surfaces and hands and all those sorts of things. So lots of things that have to be addressed to combat antibiotic resistance. Now here's an interesting thing that I thought I'd show you. First of all, let's just make sure we get what we're talking about with the development of the resistance. So this is a little population of bacteria here, and the orange ones are bacteria which have undergone some kind of genetic mutation which has given them a survival advantage in that they are resistant to antimicrobials, let's say. So the orange ones have some resistance genes and the green ones don't. Now if they're present in somebody and you don't treat that infection with an antibiotic, then what happens in the end is, assuming that infection is overcome, you may have killed bacteria, may have overcome the infection, but you haven't changed the balance between the resistant and the non-resistant bacteria in the population. Now let's compare that with the use of an antibiotic. So you've got an infection and you use an antibiotic, let's say a tetracycline, a broad spectrum antibiotic, and you kill all of the cells that are susceptible, in other words, all the bacteria that are susceptible to that drug will be knocked out. What you will be left with is just the resistant cells. And those resistant cells now have had their competition knocked out. So every time this happens and you treat a bacterial infection with an antibiotic, you are selecting for the resistant bacteria. And so over time, of course, what you will get circulating and transmitted from person to person and becoming a problem is a higher and higher incidence of the resistant bacteria being the cause of the infections, because they're the ones that your drugs are not knocking out. So you are actually refining your bacteria so that more and more of them are of the resistant type. That's a good argument for not overusing antimicrobials, but if you use them at all, then that is going to be happening. So it's not just an overuse problem. But here's an interesting thing. Now, antibiotic means antimicrobials that have come from natural sources. They're produced by other living things, like molds and other microbes, to protect those microbes from other microbes. So they've been around for a long time. We didn't just make them up. We started using them, but we didn't invent them. So we have had naturally occurring antibiotics produced by all of these different microorganisms around for a long time. Now here's the interesting thing. If you look at this graph, and this grey line here is showing those naturally occurring antibiotics that are produced by, say, fungi in the soil and so on, you don't get resistance to those antibiotics, and they are still active against the things that they were used to be active against. They've been around for tens of thousands of years. There's no evidence that there's an increasing resistance of the microbes that they were intended to fend off occurring. But yet, if you compare that with our use of antibiotics clinically, which is the black line, and we've only been using them for 50, 70 years, less than 70 years in large numbers, but look at the rate of increase of resistance. So how come when bugs produce their antibiotics themselves and use them, there doesn't seem to be a development of resistance in the microbes that they are using them against, whereas when we use them there's a huge increase in the resistance of the microbes? What's all that about? So essentially, when we use our drugs, here's a seesaw diagram, the sensitive bacteria are knocked out, leaving the way clear for the resistant bacteria there. But naturally occurring in the environment, that doesn't seem to happen. So if you have these naturally occurring antibiotics produced by mold and so on, yes, they do nobble those sensitive pathogens, they leave the resistant pathogens alone, but they don't seem to change the balance between the sensitive and the resistant pathogens. In other words, they don't do what we've done, which is selected for resistant pathogens. There seems to be something which maintains the balance. So there's some other factor which acts against the resistant pathogens, which tends to keep the whole thing in balance, and that we don't have, or we don't know about in our clinical use of antibiotics, because in our world we have got this situation. So that's an interesting thing, and it's part of what is going to need to be investigated now, that we need to look at how to wind back this resistance. One theory that has been put forward for why in the environment antibiotics do not cause resistance is that the naturally occurring antibiotics tend to degrade and form degradation products, and those degradation products in many cases actually have more action on the resistant bacteria than they do on the sensitive bacteria, so that it's almost as though the antibiotic substance has a different spectrum of activity to its degradation product. So it's almost like the naturally produced antibiotics act like a combination of drugs. That's a fascinating idea, isn't it? Now, of course, we wouldn't get this same effect using our drugs clinically, because the drugs are put in and they're washed out and they're metabolized and they're excreted and they're got rid of. You don't really get much chance for those degradation products to remain in situ and work on the resistant bacteria. So that's an interesting idea. So let's just look briefly at how bacteria achieve the resistance that they get against the various antimicrobials that we use. There's a number of different mechanisms that they seem to be able to use to evade our antimicrobial drugs. The way that they do it is by acquiring genetic material. They acquire genes which confer resistance on them, and Bacteria are very quick at acquiring genetic material. They can pass material between themselves very easily, much more easily than we can, so they don't have to wait for a whole generation of bacteria, which isn't very long anyway, but they can pass resistance genes between themselves readily. So what kind of mutations do they acquire which give them resistance? Well, first of all, the ability to express drug efflux pumps, and these are active transport mechanisms which get put into the membrane and which are able to grab drugs that have made their way into the bacteria and pump them straight back out again. Now, the bacteria may have some of these, but when they become resistant, they may have acquired a gene which allows them to make different ones or express more of those drug efflux pumps in their membranes, and therefore they have much more effective pumping mechanisms to get shot of the drug before it can do any damage. So that's one major area. Another is the production of enzymes which alter the drug in some way so that it isn't as effective. A third area is altering the drug binding site within the bacteria, or protecting the drug binding site so that the drug which would normally have its effect by binding to a particular area now can't reach that area, or the drug no longer fits the binding site, so it makes the drug completely useless. And then also, as we've seen with beta-lactamase, the bacteria acquire the ability to produce an enzyme which completely destroys the drug. It breaks the drug down so that it's no longer effective. So enzymes which either destroy the drug completely or alter the drug sufficiently that it doesn't work anymore, altering the site where the drug needs to bind, or just flat out pumping the drug back out of the bacteria. These are all ways which we know bacteria are able to gain resistance to our antimicrobial drugs. Clever little devils that they are. This is just a diagram which shows you how many different ways bacteria have of passing on those resistance genes if they acquire them. See, they can just kind of come up to each other, and the process is called conjugation. They kind of just snuggle right up to each other and attach to one another, and they can actually pass over genetic material in the form of what is known as plasmids, these little circles of genetic material. So you can say, oh, look, I just acquired a really good resistance gene. Do you fancy trying it out? Oh, thank you very much. And it's done. And you've now conferred resistance to the next bacteria. Also, viruses that infect bacteria, bacteriophages as they're known, can transfer genes, because if this virus here has infected a bacteria which has a resistance gene and it's conferred the resistance on that bacteria, it can then transfer it to another bacteria. And also bacteria have this habit of slurping up DNA from other dead bacteria which have lysed or split, and they can also take in genes that way. So they have this amazing ability to collect genetic material. We can't do any of those things. And so it's very easy for resistance genes to be passed on to bacteria. And of course they have such a rapid rate of division, many of them, that they can go through many generations in a short space of time. Anyway, this is a rather fuzzy table. I've put it in here simply to remind me to tell you really how many types of microbes are now showing resistance to the antimicrobials that we have. This is quite a scary list of bugs that are now becoming consistently more resistant to our frontline antimicrobials. So Enterococcus, these are normally part of the gut flora, Enterococcus, but they can cause septicemia if they get into the blood. They can invade surgical wounds, and whereas they used to be susceptible to a whole range of our antimicrobials, they are now resistant to aminoglycosides, cephalosporins, erythromycin, penicillins, tetracycline, and some of them to vancomycin, which is this, what I've put in brackets here. VRE stands for vancomycin resistant Enterococcus. In other words, this is Enterococcus that is now also resistant to the last drug on the list. So that's one. Haemophilus influenzae, a relatively common cause of bacterial meningitis and respiratory infections and ear infections, resistant to chloramphenicol, to penicillins, to tetracycline, to trimethoprim and sulfamethoxazole, in other words to the sulfonamides and trimethoprim. Again, multi-drug resistance for what is quite a nasty bug. The bacterium known as a mycobacterium that causes tuberculosis, multi-drug resistance is now developed. So aminoglycosides, it is resistant to ethambutol, isoniazid, rifampin. These are drugs which have traditionally been used successfully to treat tuberculosis, and we're now finding that the Mycobacterium tuberculosis has developed resistance in many areas to those drugs. Neisseria gonorrhoeae, bacterial cause of gonorrhea, now also resistant to penicillins, to tetracyclines, and to other antimicrobials, expectinomycin. Plasmodium falciparum, this is not a bacterium, this is a protozoan. It's the protozoan that's responsible for malaria, and it is resistant to chloroquine, but there are still antimalarial drugs that are effective, so we're not in such a bad way with malaria. And the list goes on. You can see there's Staphylococcus aureus. Another big problem in healthcare settings, post-surgical wounds, etc. MRSA stands for, as I said, methicillin-resistant Staph aureus, and there are now strains of Staph aureus that are known as VRSA, in other words, vancomycin-resistant Staph aureus. And Strep pneumoniae, also a cause of meningitis if it gets into the blood. There are strains of that bacterium which are now resistant to a whole list of antimicrobials. This is just a map, one of these public health maps, which is showing the areas where you're most likely to see multi-drug resistant tuberculosis infection. So as you go from pale yellow to the darker colours, you're getting a higher and higher percentage of drug resistant tuberculosis cases. So you can see that Asia in particular, and some parts of Europe, have got above 50% occurrence of multi-drug resistant tuberculosis. In Australia, we still have a relatively low, around about 10% probably, but you can see that various parts worldwide you've got a very high rate of multi-drug resistant tuberculosis. And it's a similar picture for various different bugs. This is the Clostridium difficile. Here is a map of the quinolone resistant strains of this bacteria. So the areas that are shaded in purple are showing the areas for which the quinolone resistant C. difficile strains have been reported. So it's pretty much United Kingdom and throughout Europe, most of North America and Australia. This is another map showing the occurrence of MRSA. The orange areas are the highest occurrence, so between 25 to 50% of cases are now methicillin resistant, so vancomycin then would be your only resort, and that includes Australia, many countries in Africa, United Kingdom, we've got Italy there, all sorts of places, large parts of Asia. You can see that interesting, very low incidence in Scandinavia there. That's quite interesting. But there are these maps for a whole range of antibacterial resistant strains of bacteria. So if you wanted to look, you could probably find all sorts of different bugs for which public health organisations are tracking the occurrence of resistance. This is a little timeline which is showing you a part of the problem. This is showing when various groups of antimicrobial drugs were produced, and you can see that from the sulfonamides, which were some of the first ones to come in, the beta-lactams around late 30s, 40s, just before the Second World War, then the tetracyclines. So pretty much all of the drugs that we have talked about were introduced between 1936 and 1962. We've been using them for a long time, and we haven't had new groups of antimicrobials introduced in the intervening years in great numbers. So this was really the golden age of antimicrobial production. Now, of course, we've had later generations, and there's been a steady stream of other members of these groups. But when you look at this timeline, and then you look down here, 2005, 2008, we've had a very patchy introduction of new antimicrobials, and very few new antimicrobials when you compare it to the huge explosion of antimicrobials very soon after they were discovered, and then kind of everything went quiet in antimicrobial research. And of these new drugs produced since 1999, actually only two of them, the daptomycin and linezolid, the oxazolinidones, are actually acting by different mechanisms. These other ones are just new agents in some of these older classes. So only actually 2 agents that act slightly differently from all of the groups that were developed all that time ago. And this is perhaps a little bit of a dramatic graph here, which is showing the perfect storm. And what they're talking about is, while the research and the numbers of new drugs coming onto the market is steadily declining, so the resistance to our existing antibiotics in the number of bacteria is steadily increasing. So we've got the conditions for a perfect storm. In other words, we have not been paying attention to producing more antibiotics. Between 1999 and about 2011, the number of drug companies that were actually trying to find new antimicrobial drugs dropped from 18 to 4. That's a huge decrease in interest in research in antimicrobials. We now need to pick that interest back up. We need to find new targets, we need to find new drugs, not just new members of existing classes, because if you add a new member to an existing class, the chances are that if a bug is resistant to that class, it will be resistant to a new member of that class as well. So we need to find new targets and we need to develop new antimicrobials. And we've left it very late. So that's the antibacterial resistance story, and it's not a pretty picture, but it's something that we can address. We have to get back into our labs to do so.